Hello everyone, welcome to my channel, and today I am going to discuss about the Abo blood groupings. Basically, Abo blood grouping system is a system which is developed by or discovered by Landsteiner to find out the blood groups present in the human body. According to him, the agglutinogens or antigens are present in the RBCs, and the agglutinins are present in the plasma or serum. According to Landsteiner law, if a corresponding antigen is present in RBC of a person, the same antibody should not be present in his plasma or serum. And if a person does not contain any corresponding antigen to the RBC, he must contain the corresponding antibody in the serum. So that is Landsteiner law. Now, what is Abo? Actually, he divided the group according to the presence or absence of some antigens. There are two basic antigens, antigen A and antigen B, and these are present basically in the surface of the RBCs. Corresponding antibody, this is alpha antibody, and beta antibody is present in the plasma or serum. Now, in case of the group A individuals, they have the A antigen in the RBC and beta antibody in the plasma. In case of B group individual, they have the B antigen and alpha antibody. In case of AB, they both have the A and B antigen but do not have any antibody. In case of O individuals, they do not have A or B antigen but they have the alpha and beta antibodies. Now, how it is done means how it is checked. So it is basically done, blood grouping is done by a process known as agglutination. Agglutination means clumping of RBCs like masses. So for that, the blood of an individual to be tested is mixed with 0.9% saline, and with that some test reagent such as anti-A, which consists of basically alpha antibody, and test sera anti-B, which consists of basically and beta antibody is taken. Now one slide is taken and one drop of blood to be tested is placed here, another drop is placed here. Now anti-A serum is given here, anti-B serum is given here. Now if we see, that is agglutination is in both with anti-A and anti-B, it is AB because it consists of both A and B antigen in the blood. That's why the agglutination occurs. Now we can check the agglutination either by naked eye or under the microscope by keeping the slide 2 minutes as it is, and then check it by either naked eye or in the microscope. We can find out that. Now if agglutination occurs only with anti-A or alpha antibody, it is group A because the blood consists of A antigen. If it is the agglutination occurs with the B reagent, test antisera, then it is group B. If agglutination does not occur with any of this, it is group O. because in case of group O, both A and B antigens are absent. Now it is a necessary procedure in case of blood grouping or blood matching. So in case of blood grouping, the O group blood can be given to any individual because O group does not contain any antigen and it will not react with the plasma of the recipient, and AB group is the universal recipient because they contain both A and B antigen but no antibodies, so there is no chance of any agglutination reactions if blood of another group is given to them. Now in case of blood transfusion, matching is done, that is, this is the process known as matching, that is done with the RBC serum, RBC suspension, which is prepared with the blood of the individual with normal saline plus the antiseras are taken. And in case of cross matching, the RBC of the recipient is mixed with the plasma of the donor. The RBC of the donor is mixed with the plasma of the recipient because the RBC of the donor is considered because it consists of antigen, and if we give that blood to the recipient who has the corresponding antibody in the serum, it may lead to agglutination or clumping, because in that case the body will treat it as a foreign antigen, and for that some immunological reactions will occur which will basically destroy the RBCs. And in case of blood donation or blood transfusion, the donor's antibody or plasma is not considered because the amount of blood given is relatively small as compared to the blood of the whole body, so it gets diluted and no reaction takes place. Now this A and B blood group, it is maintains the Mendelic genotype, and they are basically depends on the gene which is on the gene of the parents of the individual. So A gene and B gene is dominant and O is recessive, and the agglutinogen starts appearing after 6 months of fetal life, and at birth it is 1/5 of the adult concentration, which reaches to the adult level at the time of puberty. But the agglutinins developed only after 2 to 3 months of birth, but not during the fetal life. Now during blood transfusion, matching and cross matching, what I am saying about that, matching and cross matching should be done to avoid some transfusion reactions. Now what are the transfusion reactions that may take place? First of all, the RBC of the donor will be hemolyzed if it reacts with the corresponding antibodies, because the antibodies will adhere to the RBCs and they will destroy the RBCs, which causing hemolysis, that is excessive hemoglobin formation will take place, because hemoglobin will liberate from the destroyed RBCs, and this will increase the bilirubin concentration in the body, because hemoglobin converted to bilirubin and biliverdin, and when the level reaches 2 mg per ml, then 2 mg, in that case jaundice occurs. There may also be cardiovascular complications like cardiovascular collapse or congestive cardiac failure, because the viscosity of the blood increases due to liberated hemoglobin, which will increase the workload on the heart, as well as there will be decrease in the arterial blood pressure, which may lead to shock. There may be renal shutdown because the toxic substances released from the excessive hemolysis of RBCs, and the hemoglobin will try to get filtered out through the kidneys, and which will ultimately obstruct the kidney tubules, and which ultimately stops the formation of urine, which is known as anuria. And if this continues for 10 to 12 days and the process is not reversed with artificial kidney, then the person may also die. So this is a brief discussion regarding the Abo blood groupings and what will happen if the blood groups are not matched during transfusion or blood transfusion. And here I am ending my today's video. So in my next video, if you guys like my videos, please subscribe the channel. Thanks for watching.